Okay, Vase, what have you got from this? Hmm. 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 So amylase won't work when T is present. Okay. And when only amylase is present, there is absence of T, then amylase work. Exactly, it does work. So this means that it has changed to the reducing sugar. So that's why the iodine test is showing the negative test. Is it so? Okay. Then what is AT? Okay. So it means yes, it has some effect. So the last two one in the last two, like later in the time, that it would convert, but it has some effect on it. It delays the working of amylase enzyme. Okay. Okay, the student needs to identify with when all of the starch had been broken down in the test tube. Describe how the student can tell if the starch has broken down. Just now we have discussed. It will stay yellow brown. Okay, stays yellow brown. Okay. Next question is include in your table the time taken in seconds for starch to be broken down in all three test tubes. If starch remained after testing with all 10 drops of iodine solution, record this as greater than 270. Okay, let's make a table. How would you make a table? So there would be majorly two columns, time and then test tubes. Okay, and what should we write for A? What? How much time is taken by A? Yes, yes. Let's say it's 1, 2, 3, 90 seconds. Oh, sorry. After every 30 seconds, na? So it would be 90 second. 1, 2, 3, 4, 5, 6, 7, 8. It would be 240 seconds. And the last one would be more than 270. Right? So we have to record this in our table. So let's make two columns. One column would be of the test tube, let's say, and then the time. Test tube A, AT, and T. In A, we would write, okay, units would be seconds. A is 90, B is 240, and it is more than 270. Right? Okay. We'll discuss the paper later that was given as a mock exam because you guys have made a lots of mistakes. Lots of mistakes. Okay, what can be concluded after the effect of T on amylase activity? So the time is 240 seconds. So what is your conclusion? Good. Slows down which enzyme? Be very specific while writing the answer. Good. The T will slow down. Slow down. Slow down. The amylase activity. Okay, what is the independent variable? We won't write amount. What are we going to use the word? It's not volume. Good. This was the same mistakes you guys have made in exams. When it's volume, you need to write volume. Don't say amount. When it's mass, write mass. Okay, give a specific unit. Volume of tea. Okay, state one variable that was kept constant in this investigation. So this was the whole experiment. What was kept constant? Okay. Temperature. Okay. The sampling time or equilibration time. What else? Okay, temperature. Starch, good. Volume of starch, the sampling time. Okay, you can also write that. Okay, test tube T did not contain amylase and was used as a control. Suggest a reason for including control in an investigation. Okay, so how do we define control? For example, if we are experimenting something and we exclude that thing or exclude the independent variable, that would be our control. For example, if we are investigating the uh, effect of light intensity, excluding the light intensity would be our control, means the darkness would be our control, right? If we are checking the effect of activity of some enzyme, excluding that enzyme because enzyme is that at that time would be not present, it means the independent variable is out of it. Okay, so excluding the independent variable would be our control. So why do we use control? Good, good. To find out the effect of, find out the effect of, effect if, good, amylase is not present. Okay, explain why the all test tubes were placed in water bath. To maintain the temperature. Good. To maintain the constant temperature. Are you using key? uh, why do I have a feeling that you are using to maintain because you were about to use the word equivalent. Constant, equal, means it should be at equilibrium. A student stated that there is an error in step number 4 and 5. Identify this error and suggest how that can be improved. Let's move to step number 4 and 5. Okay, they have taken, listen, they have taken this volume in a test tube. Okay, how this uh, correction can be done, how can the accurate volume be measured. Volume, like they are taking volume in test tube, so it means this is an error. Okay, wait, what is the most accurate way of measuring the volume in bio? Exactly. So we are using a test tube, it won't give us the equal or the same amount or the same volume. Okay, this is our error. So test tube won't give the, wait, where is it? Wait. No, 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 no. Um, what what was I going to write? What what did I tell you? Uh, Test tubes. The volume in test tubes is not same because we are using test tube for like measuring the volume. It's not same. Okay. And how this can be improved? Yes, we can use measuring cylinder, but what else can be done? Because taking a 2 cubic centimeter won't be easy to adjust in that measuring cylinder. If we can reduce volume, yes. Yes, we can. We can. So we can add in water to have a perfect volume? Uh, okay. Oh, yes, we can use water to dilute it. We can. Okay. Okay. Next question. Describe a test which would show that reducing sugars have been produced. No, which would show. You have to tell. Yes, you have to tell the result as well. The process and the result. Benedict. Wait. Add Benedict solution. Benedict's solution to the sample. Okay. To the test tube, let's say. Then, yes. So this is description. So we have described it. If they have asked show how the test shows, then you have to give the results. Okay? Here we have to give only the description of test. Next question, draw the leaf. Okay, while drawing the leaf, keep few things in mind. Number one, the size. Number one, size. Number two, shape. Number three, midrib. Number four, wavy margins. Wait, there is one student waiting. Let me add. Who's this? Adina. Okay, added. Okay, so we have what we were discussing. The size should be more than the image. Number two, the wavy margins. Number three, midrib. Number four, the outline. Okay, this would give you 4 mark. Okay, and you have to draw veins. Yes. Sorry. October November 24 variant 3 P6. I've shared in the group. Oh, you are not in the group. Okay, next question. Plan an investigation to determine the effect of optimum temperature for a biological washing powder. Yes, yes. It would be similar to like how do you test an enzyme. So how would you plan? Now you are investigating the temperature. So your temperature would be your independent variable, right? So you would say that at least two to three different temperatures were investigated to check its effect or to check the effect of biological, its effect on biological washing powder. Okay. Okay. Yes, good question. How do we observe the working of washing powder? By using some stained cloth, whether the stain would be there or gets removed. Okay. Okay. How do we maintain the temperature? By water baths. Thermostat what controlled हो सकता है. Then water bath use कर सकते हैं. What else? Okay. What would be your method of washing? You would have to give the similar time. Exactly. Okay, you are not using a machine, washing machine in labs. Okay. Similar stain. We have to use the similar stain. We have to use the similar washing powder. We have to use the same concentration of water. Oh, sorry, washing powder. We have to use the same. We have to maintain the same pH. That would you maintain if you are taking the same water. So from whatever water you are using, you have to maintain the pH of that. Okay. And okay, type of stain should be same, and even the quantity of stain that you're putting on should be same. Like if you're putting on some oil, then it should be, the volume should be same for all stains. Okay. Or if you are putting on some blood stain for checking the activity of protease, then you have to put the same volume of blood on each cloth. Okay. Okay. Then you have to repeat the experiment to identify any anomaly. Okay. Okay, what can be safety precaution? Because you are exactly, exactly. Good, good. We should be careful that we should not burn our skin. Okay. Moreover, the experiment should be written in passive voice. No IUV they. Okay. Okay. The next question is related to the photograph of a villus from one part of the small intestine. What is the length of AB? I guess this is the size of paper for me. So its length is, wait, let me use my scale. Okay, it's almost 3 centimeter. If I guess this is the size of paper. So it's around 3 centimeter, or I guess 3.1 or 3.2 centimeter for you. Okay. Okay, ji. Calculate the actual width. What is the formula for actual width? Actual width is equal to image size over magnification. So if it's, let's say, if it's 3.1 centimeter is 31 millimeter, divided by 210 is 1 by 4. No, not 1 by 4. 3 into 5, 3 into 7, 1 by 7, I guess. So that would be 1 by, yeah, around 0.15. Okay, then it's written two significant figures. Okay, this is one of the word that you guys miss. Whether it's written whole number, two decimal places, two significant figures. Be very careful what is written over here. Okay? Yes, yes. Very, very bad. I don't even want to discuss. I've told you that I've checked. You can check it on Friday, but you guys didn't. Yes, yes, yes. You got 27 in, you got 26 in P6 and 30 in P2, 40. Yes. A- And P2 you got 30 out of 40. Yes, it was really poor. Very, very. I was not happy with your papers. Yes, your paper 4 was better. Because you guys even didn't bother to look at the question. You guys were so confident that the answer would be this. Uh, yeah, you must come to school someday and check the paper. Yes, I'm telling you the truth. Vase got the highest marks, 30. Okay, one question is related to graph where it's written show your working. Your working is missing. So the working was missing, and then there was a, okay, there was a question related to um, which question? There was some question related to that first question where you have to make a table, and then that the last part of that question. Most of you have got 0 marks in that. Yeah, last part of that question. Question number one. The very last part of question number one. Let's finish this first. Okay. State two ways the Villus differ from the Villus N 2.1. Uh-huh. What is the difference? I have no idea. I have no idea. I haven't calculated the percentages yet. I didn't calculate it yet. I haven't calculated. I haven't calculated yet. It has the highest percentage, right? Okay. Tell me the difference between these two. Between this diagram and this diagram. Okay. So, are you talking about the microvilli or what? So, which has a smaller surface area? 2.2. 2.2 or 2.1? 2.2. And the shape of 2.1 is rounded or pointed? Has more gaps. 2.1 has more gaps, is more smooth, while 2.1 has more gaps. Look at the diagram. It has more gaps. Okay? Okay. It's more rounded at the top. Okay. It's more rounded at the top than as compared to 2.1. Oh, why I'm using mouse. Okay. What else? What about the nuclei? Look at the nuclei over here and here. Okay, which has more black dots? One or two? Two. Two has more. Two differences are enough. Okay, let's move to the next. The diagram, okay, the graph is given. You have to draw a graph. Okay, identify the dependent variable. Okay, this is the experiment. A scientist studied the length of small intestine in different animals, the body, and recorded these things: the body length and the length of small intestine. For each animal, the length of small intestine was divided into body lengths. Okay, and the chart was given. Identify the dependent variable. Okay, by changing the length of small intestine, what would be affected? So what would be dependent? Wait. One is independent and one is dependent. Okay, what is dependent given in the question? That is independent. No, dependent. Length divided by the body length, whatever is given in this column, this one. Just wait. The horse used in the study had a body length of 2.1 meter and a small intestine length divided by the body length was 100. Calculate the length of its small intestine. Sure. Okay. One more thing. Many students have missed the working. Okay, here working is not written. But if working is written, you need to show the working. There are 2 marks of working and you guys are missing the working. Yes. You guys are continuously missing the working part. Good one. Okay, let's have this. Small intestine length divided by the body length for horse is 12. Okay, so the overall length is 12. Okay, it's written 12. Uh, the body length is 2.1 meter. What is the unit over here? The unit here is. How do you know it's meter? It can't be meter. It's not meter. It can't be meter. I guess. And how many are present? 100? 100? 100? Okay, do it then. I don't think so. It would, this would be like, it's so confusing. Wait, 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 wait. The small intestine is always longer than the body length. That is for very sure. Your small intestine is longer than you. So this must be meter. This must be meter. It can't be centimeter. Yes. Wait, let me check if it's written in the question because I think it would be meter. Um, then it must be meter. Meter, let's say meter. 2.1 into 12, greater than 24. 24, 24 and 1.2. It's 25.2. Okay. Describe the relationship between the small intestine length divided by the body length and the diet of an animal. If an animal is eating this, and this, what is the effect uh, on the length? Okay, let's say small intestine length divided by the body length is more in the animals which eat or which feed on the plant as compared to the one which feed on animals. Okay, okay, repeat it then. The animals which feed on the plants have longer, okay, than the one who feed on animals. And what about the animals who are feeding on both? They also have lesser omnivores basically. The omnivores also have the lesser length of small intestine as compared to the one who are feeding on plants. Okay, give both relationship because it's of 2 marks. First compare animal and plant one, mean the herbivore and carnivore, and secondly compare, why are you laughing, omnivore and the plant ones. I don't know. Suggest why the scientists divided, okay, why they divided the length. Thank God I was about to tell you why we do this, but I skipped that part. Why do we divide it? To have a ratio of each animal because every animal is of different size or length. So to get a ratio, like how much percentage of a body is this part, we do this. Yes, I have all of my gadgets. You should be impressed. And I'm also recording the lecture. इसीलिए मैं घर पे काम करना पसंद करती हूँ. ओबीएस ओबीएस. जी. एग्जैक्टली exactly. जी. क्या करना है सर? ये लिखा तो हुआ है. अजीब. सर, मैं लिखती हूँ. जी. Let's move to the next question. The next question is, state two possible improvements for this method. Okay, repeat. Then use use more animals or study more animals or include more animals in the study. Okay, greater the sample, bigger the sample, better the and all animals should be of same age. Okay, same diet. Okay, let's finish it.